Hello everybody, in this video lecture we will cover meiosis. So this is a PowerPoint that I will use. Let's go ahead and begin. So sexual reproduction depends on meiosis and fertilization and produces offspring that contain a unique combination of genes from the parents. So if you look over here, shows you two parents. Human are diploid organism, but inside testes and ovaries, different type of division happens that called meiosis, and sex cells are produced. Those sex cells are ovum, ova, and sperms, right? So sperm and egg must have one copy of DNA, only 23 chromosomes, because humans have 23 pairs of 46 chromosomes. So now how you go from 46 chromosomes to 23 chromosomes? And why do you need 23 chromosomes? So first you need 23 chromosomes in sperm and egg, because when they combine, when sperm fertilize an egg, then we double the DNA. 23 plus 23 give us 46 again. So we prevent doubling of DNA every generation, right? So first we reduce number of chromosomes, and then through fertilization, we get back to a normal 46 chromosomes. Now, a type of division that's responsible for reduction of ploidy, or number of chromosomes, is called meiosis. So humans are diploid organisms with body cells containing two sets of chromosomes and haploid gametes that have only one member of each homologous pair of chromosomes. In humans, a haploid sperm fuses with a haploid egg during fertilization to form a diploid zygote. So again, humans, people, 46 chromosome. Now, through meiosis, the number of chromosome is reduced to 23. So each cell over here, egg and sperm, carry only one pair, only one set of chromosome, 23 chromosome, 23 chromosome. Then they fuse through fertilization, and zygote is diploid again. And then this zygote gonna divide through mitosis to grow into new organism. So meiosis produces haploid gametes, which keeps the chromosome number from doubling every generation. To achieve the reduction in chromosome number, meiosis consists of one round of chromosome duplication and 2 rounds of nuclear division. So chromosomes are duplicated one time, but cell is divided twice, right? So if you look over here, that's a father cell, a mother cell, and first division, 2 cells are formed, and it's already, they already have half of the genes. I would say half of the genes, half of chromosomes. So we go from 46 to 23, and then each one divides again to make 4 sperm, or 4 cells, if we're talking about ovum, but 3 of them will not be functional eggs. They will be what we call polar bodies, so non-functional cells, but still 4 cells. So meiosis is preceded by an interphase consisting of G1, S, and G2 phase, the same as mitosis, which are nearly identical to the phases preceding mitosis. Right, so that's a cell cycle for meiosis. If we also have G1, S, and G2, when cell is growing, replicates its DNA, then growing again, preparing for division, and finally divides. Right, so, but in meiosis, we will have 2 divisions, right? So it's shown over here, cell divided into 2, right? And then each one is divided one more time. So meiosis includes two successive cell division processes, meiosis 1 and meiosis 2. And phases are the same in meiosis as they are in mitosis, with exception that we have this number 1 or number 2. So we have prophase 1, metaphase 1, anaphase 1, telophase 1 cytokinesis, and then prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Meiosis reduces chromosome number by half, so it's a reduction division, and daughter cells are haploid or 1N. Okay, so here we start with one cell going through meiosis right there when we have 4. So let's see what's going on over here. So we start with interphase, right? So we don't see any chromosomes yet. Centrioles are not separated. Prophase 1, centrioles migrate, we see chromosomes, right? Then nuclear envelope dissolves. Metaphase 1. Now look here. I'll show you. Compare this metaphase 1 with metaphase 2. That's equator. What lined in equator, now this 2 chromosomes. It's a pair of chromosomes. Over here in metaphase, we just have chromosomes lining in the middle. So that's a difference. So homologous chromosome line up in the middle, then homologous chromosome split, right? And short cytokinesis, short interkinesis, because it's not really interphase. So we already started with 1. We already have 2 cells over here that enter prophase 2. Now chromosomes line up in the middle, metaphase 2, sister chromatid separates, anaphase 2, and cytokinesis again, telophase. So here's 4 daughter cells, starting with 1 and end up with 4. Unique events during meiosis 1. Prophase 1, duplicated chromosomes, homologous chromosome, pair up, forming tetrads. So what shown here in blue and in this magenta color, those are homologous chromosomes. Now, let's imagine those are my chromosomes. So that's my chromosomes that I get from my dad, and this is the chromosomes that I get from my mom. So they pair. They pairs that I call them homologous. Now, why do they have these 4, kind of like 4 parts? First of all, this part is called daughter chromatid. Oh, I'm sorry, daughter, sister chromatid. This is called sister chromatid. This is sister chromatid and sister chromatid. Why I have 4 sister chromatids? Because those chromosomes are already replicated. This replication happened during interphase, right? So that means my cell is ready to divide. So I have this pair of homologous chromosomes, and now this cell goes through meiosis, not through mitosis. Then in prophase 1, this homologous chromosome from my mom and from my dad, they get together and they hug each other. And their hug is called chiasma, right? So my dad chromosome and my mom chromosome inside my cells hugging each other, forming this hug called chiasma. Now the whole structure is called tetrads because we have 4 parts. Tetra means 4. The process of pairing the homologous chromosome is called synapsis. So they synapse together, they making chiasma, and they are called tetrads. During synapsis, non-sister chromatids may cross over at point called chiasmata or chiasma. Now, okay, so here's my chromosome from dad, this is my chromosome from mom, and you see this A, B, and C, and A, B, and C, this represent genes. So gene A, gene B, and gene C. Now, why some of them are capital and some of them lowercase? Because genes are the same, but alleles are different. Alleles are represented by letters, and if it's a capital letter, that means allele is dominant. If it's a lowercase letter, allele is recessive. For example, let's imagine that gene A is a gene for hair color. So on this chromosome, I have gene for hair color. I have gene for hair color here as well, but what color? So hair color is a gene, but brown color, it's allele. And blonde color, it's allele. So alleles are alternative forms of the same genes. And brown is dominant, right? That's why it's capital, and blonde is recessive, that's why it's lowercase. Right now, when during this crossing over, so they synapsing, right? So those chromosomes synapse, they cross over and they form this chiasmata. Now, what can happen? Those sister chromatids can switch their fragments. Now look, my original chromosome over here, from there, it was all dominant alleles. Now, whatever I'm passing to my kids will have some dominant and some recessive, or some recessive, some dominant. This is called recombination. So those chromosomes are recombinant chromosomes, and this is non-recombinant chromosomes because there is no recombination, right? So at the chiasma, duplicated homologous chromosomes swap segments through this crossing over, right? So that happened in the prophase 1 of meiosis. So homologous chromosomes come together forming tetrads. When they come together, it's called synapse. So they synapsing, forming tetrads. Then they crossing over, forming chiasma, and they can switch the fragments, right? And then you end up with a recombinant chromosome and some non-recombinant chromosomes. Crossover occurs between non-sister chromatids of homologous chromosome. The result is an exchange of genetic material between homologous chromosomes. The chromosomes that have a mixture of maternal and paternal sequence are called recombinant, and the chromosomes that are completely paternal or maternal are called non-recombinant. The crossover events are the first source of genetic variation produced by meiosis. Through meiosis, eggs and sperms are produced, and each egg and sperm is really very unique. That's why your brothers, your sisters never look exactly like you. We all are different. We are so different that out of billion of people, if you know somebody, you can always recognize this person. And why we have this genetic variation? Well, because of many processes. One of them is crossing over, right? And what it says here, non-sister chromatids exchange fragment. Because if, let's go back. If sister chromatids, because this blue part and this blue part, those are sister chromatids. If they cross over, it doesn't matter, right? Because they still have the same, I guess, alleles, right? So, but when non-sister chromatids cross over, then we have exchange of genetic information here. The homologous chromosomes are arranged in the center of the cell. The orientation of each pair of homologous chromosomes at the center of the cell is random. So now look, in metaphase, not chromosomes, but the pair of chromosomes, right? Two homologous chromosomes, those tetrads. Tetrads are arranged in the middle. And what's shown here, the blue from dad, right? They are on the left and red from mom on the right, but look over here. It can be arranged in a different manner. It can be blue and red on one side and red and blue on another side. So that arrangement is random. And this randomness is called independent assortment. Because we're gonna split this chromosome between daughter cells, how we split them is random. So independent assortment is the physical basis for the generation of the second form of genetic variation in offspring. So the first source of genetic variation is crossing over, and the second source of genetic variation is independent assortment. Now, if you look over here, now when cell divides in the middle, this daughter cell only get parental chromosome, and these daughter cells only maternal, I'm sorry, not parental, paternal, from father, paternal and maternal. So this get only paternal. So here's, let's imagine those are sperm, only has paternal DNA, and this sperm only has maternal DNA. But over here, this cell has both paternal and maternal, and the same as gametes, right? So that's our independent assortment, right? Let's see over here. Next, after metaphase, we go to anaphase 1. In anaphase 1, the spindle fiber pulls the link chromosome apart. So in anaphase 1, so look, this is our tetrads. And what is pulled apart are actually chromosomes. And you see they have some recombinant chromosomes. So the sister chromatids remain tightly bound together at the centromere. So the sister chromatids are not separated. Telophase 1 and cytokinesis. End of mitosis 1, 2 haploid daughter cells, but chromosomes are still in duplicated state. Right, so here's our interphase, prophase. Prophase, we forming those tetrads, right, crossing over occur. Now in metaphase, tetrads are in the middle of the cell at the equator. And in anaphase, what pulled apart are homologous chromosomes. So look, this cell has 1, 2, 3, 4, 5, 6 chromosomes, right? So this cell has 6 chromosomes. Now each daughter cell is gonna have only 1, 2, 3. So in meiosis 1, we already reduced the ploidy, or we reduced the number of chromosomes by half. Each of 2 daughter cells from meiosis 1 now goes through meiosis 2. Similar processes to mitosis. So meiosis 2 similar to mitosis. Prophase 2, metaphase 2, anaphase 2. In anaphase 2, duplicated chromosomes or chromatids are separated. Telophase 2 and cytokinesis, nuclei have the haploid chromosome number, and end of meiosis 2, 4 haploid daughter cells. Okay, so here's our now prophase 2. That's a metaphase 2. Now, not tetrads, but chromosomes that are replicated or duplicated are in the middle, and sister chromosomes move apart, and 2 haploid daughter cells are formed, right? 2 here, total give us 4. So here's they compare meiosis 1 and meiosis 2. In meiosis 1, we have tetrads, right? And what is separating in anaphase 1 are homologous chromosomes. In meiosis 2, what line up at the equator is individual chromosomes, but they replicated. And what pulled apart are sister chromatids. So meiosis 1, homologous pair of chromosomes are pulled apart. And in anaphase 2, sister chromatids are pulled apart. And here we compare meiosis and mitosis. So mitosis is similar to meiosis 2. That's why they show kind of like arrow over here. So all the events will be similar to meiosis 2. Everything that is unique happened during meiosis 1. In meiosis 1, when tetrads line in the middle and homologous chromosomes are split in between 2 cells, we're reducing the ploidy. So the reduction of ploidy, independent assortment, right? And crossing over, everything happened in meiosis 1. So the end of meiosis 1, 4 daughter cells, each unique, and each of these cells is different from a parent cell. In mitosis, outcome is 2 daughter cells, both identical to each other and to a parent cell. So a couple more slides. Now this is also comparing DNA synthesis, meiosis, and mitosis. Right, so in meiosis, so DNA synthesis occurs in the S phase, in meiosis and mitosis, the same. I'm sorry, yeah, this is not DNA synthesis. Those are different processes and where they occur in meiosis and mitosis. So DNA synthesis in meiosis, mitosis occur in the S phase. Synapses of homologous chromosomes during prophase 1 and doesn't occur in mitosis, right? Homologous chromosome synapse only in meiosis. Crossover only in meiosis, prophase 1, does not occur in mitosis. Homologous chromosome line at the metaphase plate. A homologous chromosome does not line in mitosis, but they do line in meiosis during metaphase 1. Sister chromatids line up at metaphase plate, happen in both metaphase 2 or metaphase. And number of genetic composition of daughter cells, 4 haploid cells at the end of meiosis 2 and 2 diploid cells at the end of mitosis. So in mitosis and meiosis, the chromosomes duplicate only once during the preceding interphase. The number of cell division varies. Mitosis uses 1 division and produces 2 diploid cells, and meiosis uses 2 divisions and produces 4 haploid cells. All the events unique to meiosis occurs during meiosis 1. Okay, so now this is the last slide. In this video lecture, we cover process of meiosis that is necessary for sexual reproduction and reduction of ploidy of a cell. Thank you for watching, and I hope it was helpful.